How to identify tissue types in anatomy lab. Are you curious about how to identify different tissue types in an anatomy lab? This is a vital skill for medical students, especially in the first and second years. Let's break down the process step by step. First, it is essential to understand the four basic tissue types in the human body. These are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue covers surfaces and lines cavities, characterized by tightly packed cells. Connective tissue supports and binds other tissues, featuring a lot of extracellular matrix. Muscle tissue is specialized for contraction and includes skeletal, cardiac, and smooth muscle. Nervous tissue consists of neurons and supporting cells, responsible for transmitting electrical signals. Next, you will use microscopic examination to identify these tissues. Tissue samples are prepared as thin sections and viewed under a light microscope. Key features to observe include cell shape and arrangement. For example, epithelial cells can be squamous, cuboidal, or columnar. You should also note the number of cell layers. Epithelial tissue can be simple, stratified, pseudo-stratified, or transitional. Additionally, look for special structures like cilia or goblet cells in epithelial tissue. Staining techniques play a significant role in tissue identification. Stains enhance contrast and help highlight specific components. The most common stain is hematoxylin and eosin. Hematoxylin colors nuclei blue, while eosin stains cytoplasm and extracellular matrix pink. Another useful stain is periodic acid Schiff, which highlights carbohydrates in magenta. This is particularly helpful for identifying structures like basement membranes or mucus. Masson's trichrome stain differentiates muscle, collagen, and fibrin, making it easier to distinguish connective tissue types. When identifying specific tissue characteristics, pay attention to the details. For epithelial tissue, look for cell shape, layering, and surface specializations. For connective tissue, examine fiber types, cell types, and the appearance of the matrix. Loose connective tissue has more ground substance, while dense connective tissue has tightly packed fibers. Muscle tissue shows distinct features. Skeletal muscle has striations and multiple peripheral nuclei, cardiac muscle has striations and intercalated discs, and smooth muscle lacks striations with spindle-shaped cells. Nervous tissue can be identified by neurons with large cell bodies and processes, along with supporting glial cells. Understanding how tissue structure relates to its function is also important. For instance, the thinness of simple squamous epithelium suits diffusion in the lungs. Dense regular connective tissue has aligned fibers that provide tensile strength in tendons. Finally, practice is key. Repeated exposure to prepared slides, guided by lab manuals and instructor feedback, will improve your recognition skills. Labeling key features and comparing tissues side by side will solidify your understanding. Identifying tissue types in the anatomy lab is a systematic process that combines observation, staining, and knowledge of tissue function. Mastering these skills will be essential as you progress through medical school.